Recording in progress.
Hi everyone, welcome to my channel Swarna Men's Biology. In this video, I will be discussing with you about the molecular basis of inheritance. So let us start. So when we see this topic, molecular basis of inheritance, we will be discussing mainly about the genetic material which is responsible for the inheritance, that is structure of DNA, as well as RNA, which is a genetic material in some organisms. And the main learning objectives which we will be covering in the entire lesson are the structure of DNA, the search of genetic material, and the structure of RNA, replication, transcription, genetic code, translation, and even regulation of gene expression, and human genome project and DNA fingerprinting. And in this video, we will be mainly covering about the structure of DNA. So coming to the DNA. So DNA is deoxyribonucleic acid, and when we see the structure of DNA, so it is double helical in nature, means there are two strands which are coiled in a helical fashion over one another. So we are here seeing two strands which are helically coiled over one another. So this is the structure of the outer structure of DNA, which is the deoxyribonucleic acid, and RNA mainly consists of single strand. DNA and RNA both are made up of ribose and deoxyribonucleotides, mainly the nucleotides. So now we will be deeply studying about the structure of this DNA, which is deoxyribose nucleic acid, okay, which is the main genetic material in many organisms. So I am saying DNA is deoxyribose nucleic acid and it contains two strands. And what are these strands made up of? Each strand contains a chain of nucleotides. So each strand is made up of chain of nucleotides. Which type of nucleotides they are? They are deoxyribonucleotides. So as this DNA it is containing a chain of deoxyribose nucleotides, we are calling this genetic material as deoxyribonucleic acid. So we will be now seeing what is the difference between DNA and RNA. Now we have seen that DNA is double stranded whereas RNA is single stranded, and there is even internally there is a difference between the sugar molecule which is present in DNA as well as RNA. So that we will be going in detail. So now here in this slide, what we have learned, DNA contains the strands and each strand is a chain of nucleotides. They are deoxyribonucleotides. Okay. So, and the length of the DNA depends on the length of deoxyribonucleotides. Here, there are 8 deoxyribonucleotides. It is different in different organisms. So, the length of the DNA is depending on the length of how many deoxyribonucleotides are present. So, in the case of human DNA, the haploid content is about 3.3 into 10 to the power of 9 base pairs, whereas in the case of bacteriophage, that is 5174, it has 5386 nucleotides. Okay, and in Escherichia coli, it is 4.6 into 10 to the power of 6 base pairs. So the length of the DNA depends on the number of these base pairs, that is the nucleotides which is present in the strand. So now when we see the structure of this polynucleotide chain, which is making up one strand of the DNA, so it is made up of deoxyribonucleotides. So and each nucleotide contains three components. So each nucleotide in the case of DNA as well as in RNA, now we are studying about DNA, it contains three components. They are pentose sugar, nitrogen base, phosphate molecule. So the nucleotide contains mainly three components: pentose sugar, nitrogen base, as well as phosphate. Three components together we call it as nucleotide. So all the three components, that is pentose sugar, nitrogen base, and phosphate, all the three, okay, we are calling it as what? Nucleotide. we are calling it as nucleotide. Okay, remember? And when we take only these two, that is pentose sugar and nitrogen base, we call it as nucleoside. Okay, only the nitrogen base as well as pentose sugar, which is made up of, making up the, that is a base pair, which is making up the nucleotide. When we see, when we take only the pentose sugar as well as nitrogen base, we are calling it as nucleoside. And all the three, pentose sugar, nitrogen base, as well as phosphate, all the three we are calling it as nucleotide. Okay. And now we can write nucleoside plus phosphate. Nucleoside plus phosphate is nucleotide, or nucleotide minus phosphate, we call it as nucleoside. Okay. Now, in the coming slides, we will be seeing what type of the pentose sugar is present and what are the different nitrogen bases which is making up of our DNA as well as RNA. Phosphate is same. Okay. Phosphate as well as nitrogen base. Okay. So, pentose sugar and nitrogen base, we call it as nucleoside. Pentose sugar, nitrogen base, as well as phosphate. All the three we are calling it as nucleotide. And here you can see pentose sugar is linked to the phosphate by phosphodiester linkage, and the pentose sugar is connected to the nitrogen base by glycosidic linkage. So the pentose sugar is linked to the nitrogen base by N-glycosidic linkage, whereas phosphate molecule is attached to the pentose sugar by phosphodiester linkage. And this is a pentose sugar. Why we are calling it as pentose? Because it is a 5-carbon sugar molecule. So this, if you see here, if you see the nitrogen bases which are making up this polynucleotide chain, there are two types of the nitrogen bases, that is pyrimidines as well as purines. So coming to the pyrimidines, there are three types of the pyrimidines. They are thiamine, uracil, as well as cytosine. This thiamine as well as cytosine will be present in the DNA, whereas in the case of RNA, the thymine will not be present. In place of thymine, it is made up of, instead of thymine, uracil will be present in the case of RNA. Where we see purines, there are two types of the purines. There are adenine as well as guanine. Both is as well as in RNA as well as DNA. When we see, again I am repeating, nitrogen bases, there are two types, purines as well as pyrimidines. There are three types of the pyrimidines, thymine, uracil as well as cytosine. In the case of DNA, it is thiamine as well as cytosine. Whereas in the case of RNA, in place of thymine, there is present uracil. Whereas when we see the purines, there are two types of the purines, whereas they are adenine as well as guanine. And when we see the phosphate molecule, where it is linked, the phosphate molecule is linked to the 5th carbon in the case of DNA as well as RNA. So the phosphate will be always linked to the 5th carbon, and the first carbon, if you say this is the first, so this is the first carbon. In the case, so this is the first carbon, this is the second one, okay, third, this is the fourth, as well as this is fifth. So phosphate molecule is linked to the 5th carbon, and the nitrogen base, whether it is adenine or guanine, that is purines as well as pyrimidines, they are linked to the first carbon. Okay, so we have seen the that is a nitrogen base is linked to the first carbon, okay, by glycosidic linkage, whereas phosphate molecule is linked to the 5th carbon by phosphodiester linkage. So this is the structure of nucleotide. And here you can see the phosphate of one nucleotide is linked to the other. So how they are linked? By the 3,5 phosphodiester bond. So if you observe here, so here, so this is the first carbon, second carbon, third carbon, fourth carbon, and fifth carbon. To the fifth carbon, okay, a phosphate group is attached. So H3PO4. Okay. So for each carbon, we know OH, OH, OH. This is O. Okay. So this is a fifth carbon. But you have to remember, so we are drawing the deoxyribonucleic acid. So in the case of deoxyribonucleic acid, for the second carbon, there will be no oxygen. Okay. So what we are calling it as deoxyribo. So here only hydrogen will be present. So see here, this is the first carbon to which the base is attached. So base is attached to the first carbon. So this is the second carbon where OH is absent, means one oxygen is absent. So we are calling it as deoxy. And this is the third carbon. This is the fourth and this is fifth. And so this is one nucleotide. So if we see another nucleotide here, again same. So this is the fifth carbon to which again phosphate is attached. Okay. So by the removal of one water molecule. So this linkage, we call it as phosphodiester linkage. Okay. And this end, as here fifth carbon is this side, no? So we call it as 5' end. And so here if you see, the here third carbon is present. So this end we call it as 3' end. Understood? Again here another nucleotide will be attaching here like this. So this is the fifth carbon where again phosphate is attached. Okay. By removal of one water molecule. Likewise, one nucleotide is linked to the other by phosphodiester linkage. And this end, we are calling it as 5' end. And this end, where the third carbon is at the end, we call it as 3' end. Okay. So here the phosphate is attached to the sugar molecule by phosphodiester linkage and the base is attached to the carbon by glycosidic linkage. So this is about one strand of DNA. Okay. So now if you see here to the fifth carbon, we know that phosphate is attached to the first carbon. Nitrogen base is attached, and in the case of DNA, the second carbon, one oxygen is less, so we are calling it as deoxy, and the third carbon is linked to the next nucleotide by glycosidic linkage. Okay. See here we are finding the difference. In the case of RNA, OH is present to the second carbon, whereas in the case of DNA, one oxygen is less. So we are calling it as deoxy. Here only H will be present. Here OH is present. Here only H will be present. As one oxygen is less in the second carbon, we are calling it as deoxyribonucleotide. So when we see the structure of a polynucleotide chain, there are two strands which are coiled over one another in a helical fashion. So here we are seeing one strand and the other strand is coiled like this. And there are purines as well as pyrimidines. And both strands are complementary to one another. So if one strand is running like this, another strand will be running like this. Okay, and both are complementary to one another. And they are anti-parallel to one another. So if this is 5 dash n and this will be the 3 dash n, here the other strand runs from 3 dash n to 5 dash n. So they are complementary to one another as well as they are anti-parallel to one another. Complementary in the sense, so each chain is made up of several nucleotides. And we know the nucleotide is made up of a base, sugar, as well as a phosphate molecule. So the bases in the case of DNA as well as RNA are complementary to one another. So if adenine is present in the other strand, there will be thymine. If cytosine is present in the other strand, there will be guanine. Means in the place of adenine, the other strand contains thymine. That is a complementary strand contains thymine. Whereas if cytosine is present, the other strand contains guanine. Always, if adenine is present, thymine will be present in the complementary strand. If cytosine is present, guanine will be coming in the complementary strand. Adenine is linked to thymine by 2 hydrogen bonds, whereas guanine is linked to cytosine by 3 hydrogen bonds. Whereas in the case of RNA, in place of thymine, there will be present uracil. Okay, no thymine is present in RNA. So this is one helix. Okay, and the pitch of the helix means that one turn is about 3.4 mm, and in one pitch there are about 10 bases. So we can say the distance between two bases in the case of DNA is 0.34 mm, and the pitch of the one turn or pitch of one helix is 3.4 nm. So the pitch of one helix is 3.4 nm, and there in one helix there are about 10 nucleotides. So the distance between the two nucleotides is about 0.34 nm. Okay, means each turn or each helix contains 10 base pairs. And this is that is adenine is linked to thymine as well as guanine is linked to cytosine. So by 3 hydrogen bonds, that is guanine is linked to cytosine, whereas adenine is linked to thymine by 2 hydrogen bonds, which we call it as Chargraph rule. And this double helical structure of DNA was proposed by Watson and Crick. Okay, it is proposed by Watson and Crick. So here we can see one strand is running from 3 dash to 5 dash, whereas the complementary strand is running from 5 dash to 3 dash, and adenine is linked to thymine by 2 hydrogen bonds, and guanine is linked to cytosine by 3 hydrogen bonds. It is in case of DNA, whereas in the case of RNA, in place of thymine, there will be uracil. Thymine will not be present. In place of thymine, the complementary strand, whereas RNA is made up of a single strand, but in place of thymine, uracil will be present. Uracil will be coming in the case of thymine. Okay, why you can have a doubt? You have doubt. I'm saying you can now raise a doubt. I am saying that in case of RNA, in place of thymine, there is present uracil, and I even said that RNA is single stranded. So when the DNA is undergoing a transcription, okay, when the DNA is undergoing transcription, one strand of DNA complementary to that RNA will be produced, okay. But, that process we call it as transcription, and complementary to the adenine in the case of transcription, complementary to adenine, uracil will be present, okay. I will be clearly explaining. Okay, now see that we will be seeing in the next slides. So, in case of transcription, what happens? Complementary to the DNA strand, RNA strand will be synthesized. Okay. So in place of adenine, if RNA strand is synthesized, uracil will be present. Uracil will be coming. Okay. So RNA doesn't contain thymine. It contains uracil. This you can clearly understand when we go into the topic that is transcription. Whereas in the case of replication, complementary to DNA strand, another DNA strand is produced. So in such a case, complementary to adenine, thymine will be produced where they are linked by two hydrogen bonds. Whereas in the case of transcription, in case of complementary to adenine, uracil will be produced where it is again linked by two hydrogen bonds. So we have seen that in case of RNA, uracil will be present in case of thymine. Okay. Now, let us take a small concept test. What is the nature of the strands of the DNA complex? There are antiparallel and complementary, identical and complementary, antiparallel and non-complementary, non-identical and complementary. Just now we have seen they are antiparallel as well as complementary. Okay. So the correct answer is A. So there is explanation also. The strands of DNA are antiparallel to one another, where one strand is running from 5' to 3', whereas the other strand is running from 3' to 5'. Okay. So this is about the structure of DNA. I think you have understood this topic. If you like this topic, please give a like, share, and subscribe my channel, Swarna Ma'am's Biology. So I will be explaining you the central dogma theory as well as the structure of RNA, and we will be discussing about transcription,
Recording in progress.
When we see this nucleotide, one nucleotide is linked to the other.